Hello everyone, welcome to Brainmap. This seminar series is co-sponsored by P41-funded Center for Mesoscale Mapping, housed in Martinos Center. It's my great pleasure to introduce our speaker today, Dr. Claire Walsh. Dr. Walsh is a senior research fellow and co-director of the MSM at H group in the UCL Department of Mechanical Engineering. Claire has been interdisciplinary scientist throughout her career, with focus on using and developing imaging techniques and analysis to validate image-based computational models of human physiology. She did her PhD in the materials and tissue compartment at the UCL, and afterwards joined an interdisciplinary team at the Center for Advanced Biomedical Imaging, where she integrated multiple imaging modalities, fluorescent microscopy, for example, MRI, bioluminescence science, CT, all of them, in order to validate numerical simulations of blood flow in solid tumors. During this time, she won an MRC Skills Development Fellowship to develop empirically informed digital tissue phantoms for the validation of computational models underpinning the macrostructural MRI. And during 2020, during the COVID-19 pandemic, she joined an international team developing a synchrotron X-ray tomography technique to investigate the effects of COVID-19 on lung microstructure. Within 6 months, the team had demonstrated the feasibility, the method with which CT would be applied not only to lung, but also brain, heart, kidneys, spleen, and they got Charles Zuckerberg Initiative award. They got an award from Charles Zuckerberg to develop the technique. In 2021, Claire was awarded the ECF Young Scientist of the Year in recognition for her work, and I believe we will have a really great talk about that as well. I would like to remind the audience to please address any questions during using the Q&A box and to raise their hands at the end of the talk, but also if you have any questions, clarifications, you can raise your hands and we will just let you talk, ask your questions. Dr. Walsh, thank you for coming today, and we are waiting for the talk. Thank you very much. The virtual stage is yours.
Hi, hopefully you can hear me all right. Thank you very much for the introduction. Yeah, so good morning or afternoon, I guess, depending on where you are in the world. So as per the introduction, I'm going to speak to you today. My talk, it's entitled Correlative HiP-CT of the Human Brain to Bridge Histology and Radiology. So I'm going to talk you through all the new technique that we developed, which is called HiP-CT, a little bit about the history of the technique, about the technique itself, how it works. I'm going to show you applications to the brain and how we've been progressing the technique and what that means to the brain. And then I'm going to show you all the different kind of projects. As it was mentioned, it's quite a large international team, and we're investigating HiP-CT with applications to the brain in a number of different avenues. So I thought it would be nice to give you a flavor of everything that we're doing to hopefully spark discussion. So I'm going to begin by just taking you back in time a little bit to January, March 2020. The COVID pandemic was really hitting the world, and a lot of the institutions, for example, UCL and the ESRF, the European Synchrotron Radiation Facility, shut their doors. At this time, a team started to form. So between March and May, sorry. So Professor Peter Lee, was approached by two medics, Dr. Max Ackerman and Professor Danny Yonick, and were asked, how can we image and understand the damage that's happening to COVID-19 lungs? Peter got in contact with Paul Taffero at the European Synchrotron Radiation Facility, and also myself, and a much larger team started to form to answer this important question. The technique that emerged from this team was called HiP-CT, Hierarchical Phase Contrast Tomography. This technique bridges from the macro clinical centimeter scale whole organs, all the way down to the micro scale, similar to histology. With it, we're able to image intact human organs. And what we initially do is you'll scan the entire organ at an overview resolution of approximately 25 micrometers. Based on that overview, regions of interest can be selected, and then these can be scanned at higher resolution. And we're able to go down to about 2 micrometer voxels, a little bit smaller in some cases, which gives us single cell resolution for certain specific types of cells. Now, the technique is a propagation-based phase contrast local tomography technique. And I'm going to explain a bit more about those particular features now. So phase contrast. With normal X-ray imaging, you would be looking at absorption, the attenuation of the X-rays by the object. With our method, we're particularly interested in phase contrast. Phase contrast comes about from the refractive index differences within the material, which will give you interference and result in the contrast mechanism. The reason that we like to use phase contrast over attenuation is that phase contrast is more sensitive by approximately 3 orders of magnitude, which increases as you increase the X-ray energy. So when you're talking about imaging soft tissues, which have very, very small density differences between individual cells and bits of different sort of tissue components, having that sensitivity is incredibly important to be able to pick up these small differences. The second part that I mentioned about HiP-CT is that it's a propagation-based technique. So this is how do you turn those phase differences into actual contrast. So propagation-based imaging is relatively simple to understand. You have your source and your sample. You get interference between from the X-rays from the sample, and you have a distance between your detector and your sample. As you increase this distance, you increase the interference fringes. So as you increase this propagation distance, the distance that the waves can propagate after the sample and before the detector, you increase your sensitivity. So the take home from this is that increased propagation distance equals increased sensitivity. So the two techniques I've talked about, phase contrast and also propagation-based imaging, are not new techniques. So why did it need until 2020 for us to be able to perform HiP-CT? And the answer is the ESRF EBS. So in 2020, the ESRF was just completing and coming out of shutdown for its upgrade to the X-ray source, making it the first 4th generation source in the world. Essentially, what was critical for us is that this meant the spot size, the X-ray spot size, had reduced. When you reduce the X-ray spot size, the coherence, the spatial coherence of the X-rays increases. If you increase the spatial coherence, you're able to increase that propagation distance and thus increase your contrast, your sensitivity. Now, this is really important. Up until this point, you could apply these types of techniques like HiP-CT, but you could only do it on samples that were very dense. So fossils, for instance, paleontological samples. The density differences in soft tissue were just too small to be able to be picked up. In order to pick up these density differences, we had to increase the propagation distance. And the only way to do that is by having a more coherent source in the form of the ESRF EBS. That was where we're at in 2020, what we published. Since then, things have progressed somewhat. So the top picture, the top small yellow arrow, is the original beamline where HiP-CT was developed, BM05, and you can see that in the bottom left picture. That experimental hutch had a maximum propagation distance of 3.5 meters. The hutch just physically wasn't any bigger than that. In 2021, we moved into BM18, which is a specially designed beamline. Paul Taffro, who you can see pictured there, has been developing and designing BM18 for almost 10 years now. And it's the longest beamline in the world. It has a maximum propagation distance of 38 meters. It has the capability to go to very high energy. So let's see how things have moved on since then. So this was a brain imaged on BMO5, 3.5 meters of propagation. That same brain we took and we imaged a few months ago on BM18. This is with 38 meters of propagation. You can see quite how profound that contrast enhancement is. Here we can go even further again, and we can see that we can image at 31.6 meters, so slightly less propagation, but you'll notice that the voxel size has come down a little bit, so 23 micrometers. Just to really hammer home that point, this was our original imaging, which was on BMO5, which you may have seen published. Here, this is BM18, the new generation of HiP-CT. And here, this is the highest resolution for a whole brain that we have at the moment. So just to see a little bit of that in all its glory in video. So as you can see here, this is at the new high resolution. You can see quite how much better the contrast is going into the brainstem there. And as we zoom out and scroll, you can see the whole of the brain and some of the vasculature there, which has been highlighted. As we zoom in here, these are regions of interest that we've scanned at 6 micrometers. And you can see as we zoom through here, and you can see inside, that's one of the ventricles there. You can see some of the white matter tracks, and you'll notice that there are a lot of these very fine white dots that we see everywhere. So this is sort of the state of the art for brain imaging with HiP-CT at the moment. Sorry. But I'm just going to take a little bit of a step back. So now hopefully you have kind of an idea of what is possible with HiP-CT. I'm going to show you a little bit and talk about how we prepare the samples for this and what's involved. So sample preparation for HiP-CT involves an autopsy, at which point the sample is perfused fixed via the carotid artery. For the most part, we have a few different sources of organs. We collaborate very closely with the LADAF in Grenoble. So this is the one of the anatomical collections of France. And we also have organs that are donated through the Hanover Biobank. What is quite useful for us at the LADAF in particular is that the organs are fixed initially via an infusion via the carotid artery, which improves our fixation. We then take the organs that we want, brain, formalin fix by immersion for a week. We then do a dehydration process. So we dehydrate the organs to 70% ethanol. This increases our contrast for HiP-CT. Finally, we mount the organs in a 70% ethanol agar gel. And during this dehydration and mounting process, we also do steps to degas the organ. It's quite important to remove the dissolved gas from within the organs before performing HiP-CT, because when organs are exposed to high energy X-rays, you can induce bubbling in the sample. If you induce bubbling, you induce a lot of artifact, you get motion in the sample. So we go to quite a lot of lengths to degas and decrease the chance of bubbling. So once the organ is prepared, we then do our scanning. So with this, we perform a scan, we reconstruct the data, we select the volumes or areas of interest that we want to scan, we scan those areas, we reconstruct, and we can do this iteratively over many, many times. And then finally, we'll go into our data analysis and data sharing parts of the pipeline. I'm going to show you just a little bit about this mounting, the middle orange stage there. So the mounting in the ethanol agar is actually a very important step, one that we developed. So in the top left-hand corner, you can see the degassing that we're doing. So this can either be via vacuum degassing, or for brains, we use thermal degassing as it's gentler. We found with the vacuum degassing, initially you can induce damage in the brains. The organs are packed with the ethanol agar mix, and this serves an important function of holding the brains physically stable. So on the tomographic stage, they're rotating, so they need to be stable. And also for finding and imaging the volumes of interest that we want within the organs, we're reliant on finding the right coordinates because the organ remains stationary. So this mounting step is really, really vital for getting good image quality. And we've also found it's a really great mounting for actually a lot of other imaging modalities as well. We've used this mounting in MRI and in CT, and it works particularly well. And the organs are stable within this mounting we've had some up to 2 years. So having explained a little bit about how we get the images, I'm just going to run through this video quickly one more time, because I want to point out to you a few of the things that we're particularly interested. So as I mentioned, we dehydrate the brains in ethanol, but we don't perform any labeling. So HiP-CT is an entirely label-free technique, contrast being solely based on that density difference I mentioned before. As a result, identifying and labeling structures of interest comes down to interpretation, understanding the morphology of the brain, understanding, the types of structures that we're expecting to see. And one of our big challenges, and something that we're really still working towards, is for performing analysis on this data, we ideally want to be able to segment specific structures of interest. For certain structures, large structures, ventricles and things like this, it's relatively straightforward. But when we're really trying to push at the highest resolution to see what can we segment, what can we analyze, this is where things become slightly more tricky. So what I'm going to show you here, this on the left-hand side is the whole brain, and you can see that in the purple region is there we go, and the purple region is a volume of interest that we imaged at 6.5 micrometers per voxel, and in the green below it you can see a region of interest that we imaged at 2.56, and this was imaging regions that we actually took in collaboration with Anastasia Indiki from the Martinos Center, where we were looking at some preliminary data, with the question being is, can we see and trace white matter tracks over long distances in the brain? So this was a region chosen. We thought it might be near the STN, and we wanted to see if we could track any of the white matter tracks that came out from here. So you can see this is that region that was in purple. It's in green here it makes it easier to see. And you can see up here on the left, this here is the one of the choroid plexus here. You can just see this is part of the hippocampus here. And we're coming back down, and you've got a little bit of the cerebellum up in the top right. This is a tract that we were looking at and quite interested in. If I go to the right-hand side now, this is that same tract with the arrow in yellow, but at 2.25. And you can really see how well we can see that tract, and we can see the fibers. You can also see here in great detail, these are some of the smaller blood vessels that are running through some of the regions. And you can also see these little dots. So I mentioned in the previous video that we saw a lot of these small white dots everywhere, and we were trying to work out what they were. We were hoping that maybe we had some sort of cellular resolution there. You can see them here in the 2.25 and also in the 6.5. Now, if I go here, what you see when we look at this in, there we go. What you can see here is some 2D slices. So these are thick slab projections from that data. And you can see, if we zoom into this box here, you see these are those white blobs that we were seeing everywhere. If you look at it in the other view, you can see here, this is that tract that we were following here. And you can really see the directionality of the fibers. You'll also see that we have a lot of this sort of dendritic looking features. Now, one thing that is challenging for us at the moment, as I said, is that the identification and segmentation of all of these features. These small blobs, as you can see here, they're approximately 30 to 60 microns in diameter. So they're too large to really be a cell body. What we have to do and what we're working on at the moment is to use correlative histology. So there's so many features within the brain in particular that it could be, you know, the parenchyma of the brain has so many different types of cells, vasculature. We have many other possibilities for what we could be seeing. And so what we're actually embarking on at the moment is we are doing correlative staining. So the brains that have been HiP-CT'd already are being sent back. We're performing a variety of different stains, and we'll perform correlation. And hopefully with the aim is to be able to really segment some of those structural interests and have sort of a ground truth definitive answer for what it is that we are seeing. Then we'll be able to segment them and ideally develop a ground truth to use for machine learning techniques. Clearly, the goal with all of this, the HiP-CT work, is to produce quantitative segmentations of the data. We've explored that with a lot of other organs that we've been performing HiP-CT with, and machine learning techniques are clearly going to be at the forefront of how we achieve this. In order to perform this machine learning, we need to develop ground truths. And at the moment, particularly with the brain, the only way that we are able to develop robust ground truth is to have correlative histology to enable us to label training data, which we can then go on to use. So there's a process that we need to follow with this before we're able to produce brain-wide segmentations of many different structures of interest. So I'm going to talk now a little bit more about some of the applications. Here, you'll see just a frontal slice through the brain of an 80-year-old male who died of COVID-19 and had a clinical history of memory loss and pre-dementia. Now, as we go through, you may spot a few things with this brain, depending on how well versed you are in neuroradiology. The first thing that should strike you is the size of these ventricles. So we can see ventricular megaly is present within this brain. Another feature that you will see coming in shortly are the hippocampi here, and these are malformed. As we progress and go further through the brain, you might also notice there's a malformation of the cerebellum. So there's an absent vermis in the cerebellum. This patient, none of these neuroanatomical features were present in the medical history. So we assume they were relatively asymptomatic. However, one of the things that we saw when performing HiP-CT, so here this is HiP-CT of that same brain. You'll see here, those are the large lateral ventricles. And as we zoom in further to the brain, you might start to see these white hypo-intense regions. As we zoom in further, and this is into our 2.5 micrometer resolution imaging, you can see these hypo-intense sort of lesions. And what you might see from this is that they appear to be sort of calcific, or at least mineral-like. Now, our working hypothesis is that these are plaques based on their morphology and the clinical history of the patient. This brain has been sent for histological verification of this. But it's a very exciting prospect for HiP-CT. You can see here that these, the distribution of these plaques within one of our high resolution regions of interest with our new imaging. So this imaging video that you can see here is actually taken with data that was produced in BM05. As I've shown you, the new data from BM18 is far more detail. And we're actually able to see these hyperintense regions and many of the other smaller features, as I showed you before, across the entire brain. So what we're potentially working towards is a segmentation and quantitative analysis of the distribution of plaques within the entire human brain. If we can do this, it would be a really potentially powerful tool. We can start to say, so are they plaques? If they are, this opens a lot of doors for HiP-CT potentially to be used as a method to validate non-invasive clinical methods for amyloid beta detection. So currently, there are several PET tracers that specifically look to quantify the amount of amyloid within the brain of patients with neurodegeneration. And there's increasingly starting to become more MRI methods for this as well. A lot of these clinical techniques would really benefit from a real gold standard validation across the entire brain. Clearly, HiP-CT is still an ex vivo only technique. The radiation doses that we put through the brains are far too high for this to ever be anything but an ex vivo technique. However, for small numbers of validation cases, which we then use to develop AI algorithms for validation and further interpretation of clinical methods, we believe HiP-CT could be a really powerful tool. I also want to talk to you a little bit about some of the other projects that we have working on the brain. So one of the features that you might have noticed as we've seen some of these movies and some of the images I've shown you of the brain is the choroid plexus. The choroid plexus is the part of the brain that secretes cerebrospinal fluid, and it's thought to be quite important potentially in hydrocephalus and several other diseases. We've had an intern, medical student intern, Mr. Yanis Walcott, with us for the last few months, and he's been working on segmentations of the choroid plexus across some of these brains, in particular looking to see if we can see correlations between the morphology or structures within the choroid plexus and the ventriculomegaly or some of the other abnormalities that we've seen in the other brains. We also, you may have seen the hippocampus in a number of the images I showed you, and the hippocampus is a important structure within the brain for a number of reasons, particularly in epilepsy, forms of epilepsy where there's hippocampal sclerosis, and where the particular, you can see here quite nicely the bands within the hippocampus. We have collaborators within the Queen Square Brain Bank who are very interested in understanding what are the histological correlates for some of the hippocampal types of epilepsy. So can we see the dispersion in some of these layers within the hippocampus for cases where there's a hippocampal malformation associated with epilepsy. Another part of the HiP-CT project that we're really excited about is our work that we're doing with Siemens Healthineers. And this is really for, as an educational tool, so medical education. These are images of one of our brains, and Siemens Healthineers have rendered them using their cinematic rendering program. The idea here is really being that you could provide digital tools to help with the study of anatomy, to potentially help with surgical training, even just understanding of various different types of neuroanatomy and the types of variations we see within the population, within different types of pathologies. So this is something that we're quite excited about. Another really important part of the entire HiP-CT program is our commitment to fair data. So our portal, humanorganatlas.esrf.eu, contains all the data that we've published, and it's freely available for anyone to download. Maybe some of you have already seen it. It's supplied in several different types of binning for ease of access, and provided with the full metadata files for every data set. We're also at the moment working with Google, and we have a lot of our data up in the cloud. We're using NeuroGlancer as a tool for interactive visualization within the Human Organ Atlas platform. This we're hoping will allow much easier interaction with the data. The data sets are really large, so the full brains that I'm showing you there are up to a terabyte and a half, and this is in a compressed JPEG 2000 format. So JPEG 2000 provides a compression of about 10 times. That type of data, there's not that many institutions and people that are able to handle them, and particularly when we're wanting to interact with medical collaborators who potentially don't have the computational infrastructure, having a cloud-based interactive tool where people can view, annotate the data without downloading is proving to be really essential, and it's something that we're actively working towards. So I'm going to talk for the rest of the talk more about correlative imaging, and in particular with a focus on MRI. So you might remember from the introduction that was given, I have previously a background in working on digital tissue phantoms for digital tissue phantoms for validating microstructural diffusion MRI. And I know there's a lot of you in the audience today that work in that field, so I thought I will show you some of our ongoing and preliminary data for this. So as I mentioned, the method that we use to mount our organs, this ethanol agar mixture, is really holds the organs very robustly and actually allows us to perform correlative imaging. So after HiP-CT, we performed this 3 Tesla T2 imaging of the same brain, and because the organ is held rigidly within the same sample holder, registration between the two is incredibly simple. You can see here, this is just the comparison for BM05 and BM18 as well. One of the kind of ideas behind performing this, as I mentioned, is to be able to validate things such as microstructural MRI or diffusion tractography, diffusion tensor imaging, and tractography. So I apologize for those of you in the audience who know an awful lot more probably about this than me, but for those that are maybe less familiar, so microstructural imaging is essentially using diffusion-weighted MRI. So that's the directed and anisotropic diffusion of water within the brain to elucidate what the microstructure of those water molecules are diffusing within. So essentially extracting features such as axon diameter or directions of tracks within the brain is something that's widely done in clinical practice. However, there are a few caveats. Voxel sizes for MRI are quite large, and the signal that you are getting from the MRI is often an average signal from the microstructure within that voxel, providing robust validation for what the actual distributions of the axonal sizes are, or the specific tracts within those voxels, is something that's really important for the development of the technique and its uptake into wider clinical practice. Particular for tractography, for instance, is used in preoperative planning for tumor surgeries quite widely, but it's very well known that there's a high degree of uncertainty related to the specific paths of the tracts. Because HiP-CT has this whole organ down to single cell resolution, providing the ground truth for these microstructural measurements is something that HiP-CT is ideally suited to do. So the basic idea, you have HiP-CT, MRI, we do our higher resolution HiP-CT scans, and you can correlate segmented features within that voxel to an individual MRI voxel. Now, in order to actually do this, there are several steps that need to be completed. So optimization of the sample prep. I've talked a little bit about it. I'm going to go into a bit more nitty-gritty now. Optimization of the scanning for each of the modalities, particularly for the MR, there's a huge range of MR sequences that you might want to use. And for the HiP-CT, there's differences that we can do in terms of energy ranges and also in how we reconstruct the data to try and accentuate various different features. The analysis of the MR, this could be diffusion tensor imaging or something else. These are well-established techniques. Then you want the analysis of the HiP-CT data. If we're talking for validation of tractography, or we're looking for orientation-based metrics. So I'll talk a bit about the optimization of the sample prep, which is something that I've been working a lot on for the last year. As I showed you before, this is the kind of flow for how we prepare organs for HiP-CT. You see autopsy, formalin fix, ethanol dehydration. Now, as I mentioned, diffusion MR, for the most part, relies on the diffusion of water molecules. The dehydration with ethanol makes any form of diffusion MR not feasible any longer. So we needed to come up with a new imaging pipeline for how we could perform the diffusion MR. So to do this, I've been working with Dr. Marco Palombo and Dr. Laurent Lamal, and we've been testing and optimizing a whole bunch of different methods for sample preparation. On the left-hand side here, you can see a brain that's been prepared in a formalin agarose. This is just a single slice of the diffusion image for the people here working on the connectome scanner. Yes, the image is very quick and dirty. We're simply performing a parameter sweep of the diffusion parameters and a very quick check to see what our optimum preparation method is. So here, the problem that you have with the agarose preparation is you can see you have the poor signal to noise. Now, often for performing ex vivo diffusion MRI, people will use a proton-free fluid such as formalin, or in this case, Galden, which is the same thing, just a different brand name. What this does is, as it's proton-free, you get a zero background, so it helps you improve your signal-to-noise ratio, and also it reduces susceptibility artifacts because it matches the susceptibility, the magnetic susceptibility of tissue. And whilst this clearly is much better for the MR, we came across an interesting problem, which is that formalin or Galden provides a very good contrast in HiP-CT. I wonder if in the crowd, anyone here who has seen this effect with micro-CT, I'd be really interested. I haven't been able to find anything in the literature about it, but we found you get a very, very strong signal in HiP-CT with it, and it seems to be nearly impossible to wash it out of the brain after you've vacuumed to get it into the ventricles. So we've tried a number of different washing protocols now, and nothing seems to be able to get it back out of the brain again. So we're currently working on different methods of optimization, and we also have performed some HiP-CT of brains in agarose, and we're developing that side of the imaging further as well. So this is kind of one part of the problem. You can see here just the simple analyses that we did to begin with. So this is sort of covers these first three that I talked about: optimization of the sample prep, the scanning we'll come to when we have optimized prep, and then analysis of the MR data. Now the analysis of the HiP-CT data I'm going to talk a bit more about. This is some work that a standard white matter atlas. Now, in order to try and validate some of the diffusion diffusion imaging I was talking about before, we want to know orientation of white matter tracts within the brain. Now, with a lot of light sheet and other kind of microscopy modalities, people are able to do tracer injections, trace very specific individual white matter tracts and validate that way. As I mentioned with HiP-CT, we're label free, and so we don't have that available to us as an option. Instead, we can try and do segmentation of individual axons and white matter tracts, but this is incredibly laborious, and as I mentioned, requires that real build-up of a ground truth data set. Instead, what we have started looking at is using orientation mapping through image intensities. And this is a method that has been used for validation of diffusion MR previously, but never with human tissue, with whole intact human brains like this. So with Dr. Gary Zhang, we chose specific regions of interest within the brain where we knew there would be fiber tracts of interest. So for instance, in region 1, there's a very clear horizontal strong tract. So this is kind of a sanity check region. Region 5 and 5.1, you have a lot of interdigitation and a lot of crossing tracts. So we chose these specific regions and imaged them at multiple resolutions. We've had a master's student, Sir Simon Burt, who's been working on this for the past couple of months and has actually just finished. And what Simon did is he has developed a 3D structure tensor analysis, which some of you might be familiar with. Essentially, you're taking the gradients, the contrast gradients in the different directions within the image, and you can do an eigen decomposition here to find the eigenvectors and the eigenvalues, which will give you the principal orientations within your image. Now, there's some parameters particularly involved with performing the structure tensor analysis, which is where kind of the interest and the difficulty lie. So typically to perform this type of analysis, you would use two Gaussians to create your structure tensor matrix. You use one first, a smoothing Gaussian, to remove noise and that would lead to difficulties in calculating the derivatives. So this is just a smoothing one. And then you have a second Gaussian distribution, which gives you your window size over which you're actually going to calculate your gradients. So your first smoothing and your second is your window size, which is the idea that this is the kind of range over which you think there might be orientation or that you want to identify orientations. So quite how you choose these parameters, especially if you're trying to validate a specific diffusion MR sequence, is not trivial. And depending on the exact sequence you use, that will affect the second Gaussian, the window sizing you use, and dependent on the imaging modality, it can affect how much smoothing that you need to perform. On the right-hand side, you can see over here, this is a small portion of data from the cerebellum that was just used as test data. And you can see in the bottom here, this is the orientation for the principal eigenvector orientations in there that we've done. Now, to show you just some very preliminary data, so presented to me for the first time yesterday. So this is some of the very first orientation analyses that are being performed on the whole brain. And here you have that region 1 at higher resolution. Now, as I mentioned, the data sets are really, really large here. So this is processing, I think this region 1 data set on its own is about 800 gigabytes. So processing this data and visualizing this data are incredibly challenging, non-trivial problems, which we are still working on. But you can see the kind of direction in which we are moving. So watch this space. I was told by Bruce that there were also some heart people in the audience, so I thought I would give you a bit of a heart teaser. HiP-CT is equally applicable to the heart, and as is the orientation type analysis methods that we're looking at here. In the heart, we can actually image all the way right down to the cardiac myocytes. So you can really look to see sheetlet orientation and things like that in the within the heart. There you see the cardiomyocytes quite distinctly, and a lot of the microvasculature. So, just to conclude, HiP-CT is a new tool for neuroscience utilizing the ESRF EBS. It enables imaging of intact human brains down to 2 micrometers per voxel isotropic anywhere, but not everywhere in the entire brain. It's a label-free technique. Contrast is based on density differences. Whole brains can now be imaged at about 18 micrometers per voxel in approximately 10 hours. The correlation of HiP-CT to MR-CT and histology could provide validation for clinical biomarkers of neurodegenerative diseases and also many other pathologies. This work is part of a huge team. I'm one in a very massive team, and I need to thank all of our funders, in particularly the Chan Zuckerberg Initiative, who are one of our great supporters of this work. And also we have a lot of resources for people who are interested to go and find out more. So we have a YouTube channel where you can go and see some of those videos. Our papers are available here, the Nature Methods paper. We also have a protocol paper coming out soon. This here is an example of our NeuroGlancer interface that we're working with, the Human Organ Atlas that I mentioned here, and our group website. So I'd just like to finish by happily taking any of your questions and thanking the huge team, in particular Paul Tafro and Peter Lee, who are the co-leads on this project.